So good afternoon, and thanks very much to Helen and the committee for asking me to speak about molecular profiling in the UK and Europe for cholangiocarcinoma patients. It's a subject very close to my heart. So you've all seen a lot of information about cholangiocarcinoma at this meeting, and we can see that it's divided up into its four main anatomical, biliary tract cancer is divided up into its four main anatomical components: the intrahepatic, the hilar, the extrahepatic, and gallbladder cancer. And we've learned how to use chemotherapy to treat these cancers. The UK has led that process. However, it is really the genomics that have become the excitement in this cancer. And this was published originally in 2015. The Japanese published these data that we see here, and it was expanded in 2017 by the Thai and Singapore groups. And this looks at all the genetic drivers within biliary tract cancer. And it's unusual in both the richness of the potential drivers and also their targetability, and I hope to be able to explain that. There's some peculiarities about it. So the intrahepatic cancers have particular drivers, particular genetic abnormalities that can be targeted, and the gallbladder cancers have other particular abnormalities that can be targeted. There are some abnormalities that are common to the whole group. These diagrams here are meant to demonstrate how some abnormalities are shared with others. And then if I can give you an example of one of the more famous ones, the FGFR2 abnormality just here. Although some are shared with this abnormality, to P53, most are shared with this abnormality, the BAP1. Nobody really understands why it's shared with BAP1 or indeed P53, but the critical thing is that it's not shared with any other potentially harmful genetic abnormalities such as RAS. RAS is a real bad guy in cancer, and it's not shared with either of those. And so that makes it a really attractive target, because if you target that, you haven't got something else that's going to spoil the show. So using FGFR as an example, FGFR is in fact translates as fibroblast growth factor receptor, and there are 4 of these, and they work by once they're stimulated, they work by driving various what we call pathways within the cell, and these pathways go into the nucleus here. So this is a cell and this is a nucleus, and they go into the nucleus and make and go into overdrive, and the cancer goes out of control because of this overdrive. And the importance of finding out these abnormalities was highlighted by the cholangiocarcinoma organization in the States. You'll see a little more of that later on, but it's really important, and to emphasize the need to look for these mutations. And here's some of the early evidence as to why it is so important in cholangiocarcinoma and biliary tract cancer as a whole. And the original data come from the Moscato study. So it's a French study, and 1,035 patients, so a lot of patients, were profiled. They had their molecular profile performed looking for abnormalities as the abnormalities some of which I've shown you earlier, looking for some handle, some area to treat. Of those 1,035, 43 patients had biliary tract cancer, out of which eventually 18 were treated, and this is what's called, so you get to quite small numbers in the end, but these are important numbers. On the right here is a thing called the waterfall plot. Anybody and each bar is a patient, and any patient who's who's over here underneath the line means that their cancers have fallen in size. And so you see that actually quite a large number, a third in fact, a third of patients have responded. And the information we draw from these preliminary French data was that not only is biliary tract cancer and cholangiocarcinoma, not only does it have more targets than the average cancer, but that when you treat these targets, they respond better. And so that's a really exciting base. As such, cholangiocarcinoma is special. It's special because it appears to differ from other cancers in this way, making it ever so much more important to look for these genetic abnormalities. Here were, if you look at these abnormalities, so these guys on the right here, the blue guys on the far right here, what drugs did they actually receive? Well, it was a bit of a mishmash of all sorts of drugs, and consequently, it's even more impressive. The result of such a good response rate is even more impressive because us doctors don't particularly like it when patients get a mixture of different treatments. We can't draw any clear conclusions. So the fact that 6 people responded is even more impressive. And these were the abnormalities which they had, and you could just read that off the bottom of the graph there. 4 of them had these FGFR abnormalities. 1 of them had an EGFR abnormality. This is an ERB2, so that's the same gene as in breast cancer. ERB3 and ERB2, these are similar genes to breast cancer. And so all of a sudden, you see why, if you look for these genes, you do find the abnormalities, you can give the drugs, and responses are obtained. So that's really exciting from our point of view. Let's focus on FGFR as an example. And there are now quite a few FGFR drugs out there, but I think the general view is they're all very similar. So if you look at these 4 drugs, infigratinib, pemigatinib, futibatinib, and derazantinib, I don't expect anybody to recite that on early in the morning. They're all of these, what we call waterfall plots, similar to the one I showed you earlier. They're all rather similar, and so these drugs, generally speaking, have a very similar effect. What about the rest of them? So we use in your average profile, we're looking these days for a number of other things in addition to the FGFR. We're looking for something called IDH1. We're looking for something called HER2, the breast cancer drug. We're looking for MSI, which is microsatellite instability. We're looking for a gene called BRAF. We're looking for a gene called BRCA, and also EGFR. If you add all of those up, approximately 50% of patients may have targets to be used, and that really is that is not only more than the average cancer. These drugs are an example of the drugs that can be used. The drugs are out there to be used. You don't not only got the gene, you've got the drug. The match is very important, and the evidence that they work. This is in fact a European scale for showing how they work. Anything 3 is very good, 2 is okay, and so there is evidence that they work. The evidence is probably not is probably best for this drug here, this ivosidenib drug, but there is evidence that these drugs, for the most part, work for these patients. And but doing the test is really, really important. And if you look for if you compare different tests and different ways of doing it, you get quite a few different answers. So if you look at the top here are our usual suspects, which I showed to you in the previous slide. And this test is done using a commercially available test called Tempus. And you see there's a big difference between doing it on the biopsy here, or doing it from a blood test, using what we call a liquid biopsy. and that and for some abnormalities, for instance FGFR, it's clearly for this test, the liquid is better, but for other ones, it's much the same. And for the IDH1, it's much the same, and that gives you an idea of what to use, what to expect if you're going to use this test. The situation is different if you use a different test. So if you use this test called Foundation Medicine, and the columns here refer to the abnormalities found in the primary cancer, in metastatic cancer, and in liquid biopsies over here. And you can see there is really quite a marked difference between each of these. So it's not a simple case, do the test, get the result. You have to think carefully about the test. You'd have to think carefully about how you do the test. You have to think carefully about whether you do it to the primary cancer or to the metastatic cancer. So all these things should come into a discussion about what test to do and in what way. The other aspect of management, which all this testing has brought up, which we haven't really gone into any depth, is how to manage patients' expectations through this extended series of roller coasters. Treatment, cancer treatment, and the diagnosis is a roller coaster at the best of times. Molecular testing adds a new level to that. And if we go through from the left here, the colors indicate the level of stress that the patient is undergoing. And you start up on the far left, it will be possible to profile my tumor. Currently, a lot of these tests are not publicly available. People have to pay or go into a clinical trial in order to get tested. Most people, it is possible, but some people it may not be possible. There may simply not be enough biopsy, or the liquid biopsy may fail. A lot of questions around there. A mutation will be found. So as I've said, about 50%. 50% of people don't have a mutation. There will be a matched treatment. Well, I think this is for some people, they'll be treatable on a study. For some people, the drugs will be available. It's yet another question mark in the process. I'll be able to access the treatment, so that's whether the treatment is actually available on the NHS. And finally, I will respond. If you, the pathway, the treatment pathway has always been difficult, and I don't think targeted therapies and testing makes it any easier. There are now a huge number of people to help us get through this ordeal. There's of course Helen and the sterling work that she's done. There's the American equivalent, the Cholangiocarcinoma Foundation. They've done a great deal to bring things to order. There's ENSCA, which is run by this guy, Jesus Benales, and that's primarily a research and scientific organization. There's Cholangiocarcinoma UK, run by Shahid Khan. That's part of the British Association for the Study of the Liver, and that essentially has an overview of the activity of cholangiocarcinoma research in the UK. There's EuroCan, for which we're now no longer formal members since Brexit, but we have a very good association with EuroCan, which is essentially a tissue collection organization. And then there's an IBTCC, which is a further organization. So all of these organizations are here to help us get this right, get the treatment, get the target, and get the treatment for that target. So that's all I had to say, except for now a cartoon which we're about to show you about targeted therapies. So this is a cartoon produced by Insight in collaboration with the AMMF. It's primarily to increase awareness about testing and how to do the testing and the importance of testing. It will be on the AMMF website, but you're very welcome to use it, show it to people, use it to make people more aware of the importance of testing in cholangiocarcinoma. It's primarily aimed at patients, but actually there are people in there. It's also aimed at physicians as well, to make them aware as to the importance of it. We're about to release it. We're about to show it to you now. It'll be available on the AMMF website. Please let us know what you think of it. And with that, I thank you for your attention.
My name's Erica. I'm 53. I've got 2 teenagers at home, and one of them has flown the nest. I work in an insurance company. I'm just like anyone else, really, except I've got cholangiocarcinoma. It's a rare type of liver cancer and one of the toughest ones to treat. My care team reckon I've had it for years. Getting diagnosed was a terrible shock to the system, for me, my husband and my kids. I'd had some pains in my stomach, but I didn't think they were serious. By the time I saw an expert, it was too late to operate. A few months ago, I didn't have any real worries, but now my life feels so uncertain. But my doctor tells me things are changing in cholangiocarcinoma, so I'm hopeful about the options that might be available to me.
Cholangiocarcinoma is a very aggressive cancer, and due to a lack of specific symptoms, it's often diagnosed late. This can make it hard to treat, and currently there are very few treatment options available for these patients.
My diagnosis came after a biopsy, like a big injection, except they take tumour tissue out instead of putting medicine in. It was just a small piece of me, but precious and incredibly important. It just may be a key to unlock new options for patients like me.
Molecular testing has become essential in the fight against rare cancers like cholangiocarcinoma. All of us have an increasingly important role to play. From ensuring enough tissue is taken during biopsy, to using it sparingly during diagnosis to make sure there's enough left over for molecular testing.
My team were great. They've made that tiny bit of tissue go a long way. And now I'm waiting. If certain genetic changes are found in my cancer, it might make me suitable for new targeted treatments. So, well, fingers crossed.
The cells in our bodies are constantly dividing. Sometimes, when certain genes in the cells change and become faulty, for example, when mutations occur, it can drive the cells to grow out of control. This is how cancer is formed. In order to try to stop the cancer growing, these faulty genes have to be brought under control. This can be done by selecting the right treatment. Almost half of cholangiocarcinoma patients have genetic changes, like FGFR2 fusions, which can potentially be acted upon. Molecular testing helps us to identify patients who have faulty genes in their cancer, and in turn, those who might benefit from targeted therapies that could significantly extend their life. And as more genetic alterations are being explored, we are creating a genetic footprint for the identification and treatment of the cancers of others in the future.
It's been a difficult time for sure. But knowing I might have a way to get a bit more time, that keeps me going. And even if I don't, it's nice how that little part of me has contributed to science. That it might help someone else like me in the future. That's something.
If you've been diagnosed with cholangiocarcinoma, ask your doctor about molecular testing.